Alright class, welcome back. We're talking about anatomy and physiology. We've gone into chapter 2. And this is going to be a four-parter video set on cells and understanding of the cell and how that works and how we're going to deal with the cells, and then we're going to start talking about them in the different structures and why that's important. And again, you can see a lot of learning objectives here, so if you need to follow along with the slides at home, by all means, that they are up on canvas so we can definitely see them. So guys, cells are the foundation of the human body, okay? And again, there are single-celled organisms. An amoeba is a single-cell organism. And then the human body contains billions of cells, and they usually have a specialized function in them, depending on, again, usually based upon function, okay? So your protoplasm is the viscous matrix that supports the interior cellular structures. It's what they float in, this protoplasm or cytoplasm. Again, each cell has a minute mass of a colorless substance, and again, it's a medium for the transport of different things. ATP being your energy molecule, and then nutrients, signaling molecules, and proteins. So again, tissues are the cells that have a common job. We talked about this last time. We were talking about chapter 1, and then the tissues become organs. The organs again become organ systems then become organisms. But again, we're constantly battling for this thing called homeostasis, where we're trying to return to a normal state, or what the cells think are a normal state. But when you have a disruption of homeostasis, you can have a serious impact on the entire organism. Kind of goes without saying. So there's two general classes of cells. Again, your sex cells and your somatic cells. And then we're gonna be focusing on the somatic ones. When we get to the reproductive section, we'll talk about not only mitosis, but we'll be talking about meiosis, where it splits at a different time. But again, mitosis is the basically cell division that we'll talk about a little bit later. There's three parts of a cell. Again, this the membrane, the nucleus, the cytoplasm. Nucleus usually contains your genetic code, the control of the cell. The membrane lets things in and out, and we're going to talk a lot about membranes because a lot of the drugs that we have work off of membranes. Okay, so your organelles are the microscopic things that perform functions within the body. Now, do I need to know all of these? My answer is, yeah, you do. But if I had to focus on a couple, nucleus is definitely one I would focus on. Definitely your mitochondria. I would definitely make sure I knew about that. Your centrioles are used for cell division. Lysosomes are kind of used to destroy the cell. Again, in the Golgi apparatus, they usually are having to do with energy and protein synthesis. So anything outside the cell, extracellular, inside the cell, intracellular, okay? And so the membrane is what controls the movement in and out. And that's kind of a big one, especially when we start talking about function. We start talking about shifting potassium from the inside to the outside of the cell and vice versa. And there's a process that usually requires energy, by the way, in order to do that. We don't have energy, we don't have that process. So your atoms, again, protons, neutrons, electrons, we already discussed these. But the thing to remember about the atoms is you can get positive and negative charges out of that, something that our cell membrane actually kind of uses to let things in and out. Again, and it detects signals from outside the cell and transmits them to the inside. They do this via the cell membrane through gated systems. It lets things in or out selectively. It can be semi-permeable, so it will allow some things to come in while it won't. Think of the membrane as the bouncer at the club. He's going to let certain things in, certain things not. And again, only substances that can enter and leave the cell. That's the whole theory behind that. So again, lipids and proteins, they make up the cell membranes, and it's usually a double layered, and this is that double layer. And remember we talked about the phosphate ions? Again, there's usually a polar head, which helps repel water, and then again, this particular layer, the tails, if you will, are usually it's a bilayer that allows it not to come into there. So usually the phosphate portion is the outer portion, and the fatty acid protein is on the inner surface. Now what they'll have is these channels that allow things in or out, and they usually they're have to do with a different protein that activates this to allow certain things in and out. Different enzymes do this, and I'll give you an example. Different hormones do this. Insulin, it would be an example of helping to carry a glucose molecule through one of these channels. So these are the things that they actually will do. So that, again, it helps to maintain homeostasis and allows some things in, allows some things not in. But this is how you maintain the balance there, okay? Which, by the way, is a little sidebar here. The reason that we love propofol so much. Remember that this layer right here, guys, is all made out of lipids, okay? By the way, it will move around to let other lipids in. Diprovan or propofol is a lipid-based drug, which is why it goes through those layers so quickly, versus our other drugs, succinylcholine, Versed, midazolam. All of these guys are water-based drugs, and of course, this layer up here helps repel water-based. So again, until it finds one of those channels, it usually will not let them in, okay? So again, it depends on what different types of things it can and cannot let in, all right? And again, that's why those channels are in place in the side of the membranes to help carry things in up to and including, let's say, glucose molecules, okay? Glucose, relatively speaking, is a large molecule. It's not going to let things in too easily. Now, electrolytes, these are chemicals made up of salts or acids. They're ionic conductors. And again, and there's several different, by the way, there's several different ways of getting things through that cell membrane. And we kind of covered the one, which is diffusion. We'll talk about the other ones here: osmosis, active transport, and then endo and exocytosis. So diffusion is the movement of particles from a high concentration to a low concentration, okay? And it's really important, again, it wants to be equal. All of these guys out here want to equalize without there, but this is where the membrane only lets a little bit of them through, or it lets them in through a channel, okay? Now, and again, it kind of depends on the permeability of the membrane, and how much of the concentration is sitting on the outside. If this is really concentrated, it's going to flow through there a whole lot easier, okay? And again, small molecules diffuse more easily than large ones, and the watery solution diffuses more rapidly than thicker viscous solutions. So like your plasmalates, blood for that matter, doesn't usually diffuse too well. Again, your thicker glucose molecules don't diffuse that well. Osmosis. Now this guy is from low concentration to high concentration through a semi-permeable membrane. Most of our cells use osmotic pressure, okay? And it usually has to do with the tendency of water. Osmosis, guys, think of water, okay? So again, crenation, it usually shrinks the cell, and then lysis is it swells the cell where it eventually bursts, okay? Facilitated diffusion usually requires some sort of carrier. So glucose would be a great example. It needs the facilitator, which is insulin, to carry the glucose molecule through the cell membrane, okay? Usually, and by the way, energy is not required for that, and the number of molecules transported is directly dependent upon how much concentration. Now, there are some times that we actually want to pump different ions one direction or the other. The sodium-potassium pump would be an excellent example of that, but it requires that of energy on the outside of it to open the channel to allow the ions in and some cases out. So active transport requires additional energy in order for that to work. Filtration is the movement of water out of the plasma across the capillary membrane. Usually, kidneys is a good area where filtration happens. Liver as well. Again, it's usually caused by hydrostatic pressure or oncotic forces. That oncotic will pull the water back into the capillaries from the interstitial space. A lot of our drugs, by the way, that work on the kidneys play with this oncotic pressure. That's kind of what it does. Now, again, active transport, again, is a movement from the substance against the concentration gradient. It requires energy for it to work, okay? So, again, it moves opposite that of diffusion. It will pull in the ions that it needs, okay? The gates uses energy to pull them through, okay? Endocytosis, where it actually the cell is uptake of material where they essentially surround it and engulf it, okay? And versus exocytosis, by the way, that's phagocytosis is what I kind of just described. But the cell can also drink the liquid of another cell or another area, which is called cell drinking. But your phagocytosis is usually the big one when we're talking about endocytosis, especially a lot of your white blood cells. The other thing that it does is with exocytosis. So usually it releases material out into either the bloodstream or the plasma. What happens is it forms a little vesicle around it towards the membrane, and then it releases that vesicle and it binds to the membrane and then just goes out and it's eliminated from the cell. And again, digestive enzymes would be an example of that. And then mucus secretion in the salivary glands, yes. While you're swapping spit, just remember that is an exocytosis reaction. Yeah, keep that one in mind next time. All right, we're going to stop there because we're going to start into the organelles, and we'll start in on the next video for that. I'll see you guys on the next one.